Hello, everyone. I'm Hannah Rudman. A very virtual, warm welcome to you. I'm the Senior Challenge Fellow and Data Policy Lead at Scotland's Rural College, SRUC, and I'm here with a number of colleagues today for this Rising to the Challenge session. So we wanted to introduce to you as computer scientists some of the big challenges and opportunities that we have within our research, and also introduce you to the challenge centres and a little bit to SRUC as well. So I'm going to do that first, and then we'll each briefly introduce our research focus and highlight the problems that we think that you as computer scientists might be able to help us solve. So there's going to be an opportunity for a kind of initial ideation session with each of the researchers presenting, and the aim is then to see whether there might be any potential solutions to the problems that we identify, and whether that's worth workshopping further, perhaps into a collaborative project with some further success support. So hi, Alan, thank you for joining the session. I have the questions from folk coming in on my iPad here, so if I look off to the side, that's what I'm doing. And I'll keep an eye on timings just to make sure that all of us are fitting in within our time slots, and that we've got lots of time for ideation. So I guess you're entirely used to the setup already, but obviously raise your hand if you've got questions, and I'll bring you into the chat. And then do feel free to use the involved system as well if you prefer just to type the questions in. All right. So let me just clear my screen so that I can see what I'm looking at. Great. About the challenge centres then that SRUC has just recently launched. Research at SRUC delivers findings to help us think about how we transform the world around us. That's locally in Scotland and globally as well. And our challenge-focused research portfolio is outcome and impact focused. And the centres that we have recently launched are driving data and digital innovation, a centre around the global climate emergency, a centre around safe and improved food, considering the thriving natural capital for Scotland, and considering future food systems as well. So of course, all those centres set up to be collaborative, to work in partnership with industry, policymakers and society. I've included this rather nice diagram. It shows how our academic departments, which are the kind of smaller little wheel in the bigger circle, how those departments kind of speak to the new challenge centres, which are the bigger dots in the bigger circle. There'll be collaborative centres, but obviously they kind of bring together our academic strengths together with the commercial and development activities that SRUC does as well. So we're really keen that we impact policy, and we're really keen that we impact knowledge as well. And I suppose I kind of see that inner pie chart as probably spinning around with each of the departments kind of adding their expertise to the centres. So the research centres really there to enable us to collaborate. And if you think that you have projects that we should be partnering on at an institutional level, please do get in touch with us. All right, so that's a bit about the challenge centres, and please, as I say, do get in touch. I'm going to now introduce my research focus and highlight some problems that you as computer scientists could help me solve. So I'm going to talk about that for 5 minutes, and then we'll go into an ideation session, and then my colleagues will share their research focuses and their problems as well. All right, so my research interest is in re-establishing trust in technology. Now, the importance of trust is really only noticed when it becomes scarce, and that's happened a lot recently. Digital trust is constantly being manipulated. 2 Financial Times investigations earlier this year revealed just how much trust can be manipulated, even on the best resourced tech platforms. So here we have Amazon's online marketplace in the UK, which the FT discovered had been infested with fake reviews, giving 5-star recommendations to little known products. And following the investigation, Amazon deleted 20,000 product reviews. And then their second investigation looked at how highly organised ratings farms have manipulated rankings on Apple's App Store. And that's an issue because, of course, boosting the rating of an app above 4 stars can have a really big impact on its sales and usage. And on a platform that facilitates more than $500 billion worth of commerce per year, it's important to keep that fair. So what new technologies can help? Well, we have been thinking about how distributed ledger technology, or DLT, can help. It's got the ability to enforce privacy and commercial confidentiality for businesses that want to work together in complex chains of custody. And DLT's potential, because of its attributes of advanced cryptography, means that it's suitable for really secure data protection. And in its private and permissioned format, we think it's probably an appropriate platform for multiple parties that want to collaborate digitally to do things together, perhaps because they have to in a supply chain or because they have a chain of custody around data. We think that that's an appropriate platform for bridging that IT and transparency and security and trust gap. At SIUC, we've been using DLT to prove certain qualities are maintained throughout product lifecycle. We've been working with innovative Scottish farmers to use DLT software to assure customers via an app that oats remain uncontaminated and gluten-free throughout their life cycle. So that's important actually if you've got a gluten allergy, because although oats are naturally gluten-free, they can become contaminated as they grow in the fields or as they go through processing. So in our project, the parties in the supply chain from the farm to the real retailer added digital data to a shared but encrypted register in a controlled and secure process in a private and permissioned network. Some of the data was given permission to be public, and the solution allows consumers to trace their oats back through the supply chain to track what's happened to them through the life cycle, so that they can be completely sure that those oats are gluten-free by that public data available via the app. The app, of course, is accessed through the QR code on the packaging, so that can just be scanned to give that dashboard of public information proving the oats journey from farm to fork. Also at SIUC, we've been thinking recently about if humans can better trust newer technologies such as DLT to kind of prove really secure permissioning and security of data. How could we use newer technologies like these to re-establish trust with citizens around the accuracy and truth of scientific data that actually helps us address some of the world's prescient challenges such as climate emergency or food security? So, of course, we're all aware of these alarming words uttered by the most powerful man in the US. People surely wouldn't follow them, you'd think. But unfortunately, a recent Pew Research report has revealed that the American public has increasingly echoed this sort of mistrust in scientific data over the last 5 years. So, of course, there's a very big difference between reasonable mistrust of scientific process and then an outright rejection of all things scientific just because it clashes with one's personal beliefs. So what if scientists could come back to the public with ledgers that showed the truth, that verified their data collection, that proved auditable validation of that data collection from other scientists or other bodies? So what if we could provide that trust in our data? SRUC's got campuses and farms and research centres and labs across Scotland, and this is our Hill and Mountain Research Centre in Kirkton in Scottish Uplands. So second bit of pink on the map that you can see, and to the left of that. And we've got sensors on that site. They gather information about soil temperature, moisture, as well as river level and air and humidity centres. So that gives us a constant stream of data. What if we could collect that onto ledgers for truer, more trustable points of data proof? And then what about sensors that we could use that people might trust around climate change? So we have been gathering data about how the mountain woodland, which is circled in red here, about how sensors there give us information about levels of carbon that the woodland and the soil is sequestering. So DLT is definitely a more trustable technology. It can tell us the truth about how the data has been gathered, and we can use that data that we are beginning to collect from the intelligent systems on our farms and labs, like the sensors at the Hill and Mountain Centre, to prove the positive impacts of how we manage the land. So that's my challenge. I just wanted to discuss ideas of how we could strongly authenticate IoT sensors, and whether we could better establish trust in technologies like DLT to help us build that human ease and trust with data about climate and food systems. So that is my challenge. How could we better establish trust in technology? So, anybody like to come up with ideas about that? I'm just going to try and see the participants list now and see if anyone has any thoughts or comments or raised hands. And then I'm also going to just check our questions on involved. All right, there doesn't seem to be anything on involved. So anybody got any thoughts in the group, please, in the participants box, you can just raise your hand and then I can see you and unmute you. Okay, well, if I could ask Vijay to just get ready and just begin to prepare to present.
All right. So it says, can you stop your
Yes,
screen
I
sharing?
will just stop sharing and then you should be able to.
So you can see, right?
Yes.
All right, great. Wait a minute. So, shall I start now
Yeah, please do, Vijay. Thank you.
good afternoon to everyone. This is myself, Dr. Vijay Kumar Gupta. I am Senior Challenge Research Fellow in SRUC, recently joined in July. So my CISCA pitch on is on application of computer LC analysis to studies to evaluate the environmental impact assessment of bio-based biorefinery systems and microbial bioprocesses. So with the very first, it's a little bit introduction about myself, the research focus, what I've been doing past few years, and the focus I'm going to do as a senior research fellow in SRUC under the two centers, Center for Safe and Improved Food and Bio Refining Center in SRUC. So far, my focus will be based on the biomass valorization and use of microbial technology to convert low value biomass or waste material to high value end products using the technology, and also extraction of bioactives from let's say biomass as well as microbial sources, and having the five research technicals platform that includes extraction of bioactives like using pharma and antimicrobials, and then biopolymers, biochemicals, biofuel, and fermented residues, which is so here we are using microbial technology as enabling key for upstream bioconversion for downstream bioprocessing to value-added products. Uh, and the second one is more about the processing technologies involved conversion of low value end products to the high value product, including chemicals, food, energy, materials, and leading towards um, zero waste or having a concept of circular economy. So here the question, and also the aim of this pitch is actually to provide to have with the help of computational scientists to provide a computational based scientific approach for life cycle assessment of bio-based refineries and microbial bioprocess. So with that in mind, we have data curation for especially for impact analysis of feedstock selections to microbial cell factory selection technologies as a two good practice for early stage assessment. And then also the second stage is about the more about the process optimization indicators selections like cost, operation, energy, and environmental factor to be used. Here I would say the key question to be addressed is how can we switch industrial process production processes from complex to a defined media. Remember, I'm saying complex processes to defined media, such as with that, computer-based approaches can be used to reduce a lot variability, environmental variability. I will come to the example as a bit, and then stability and environmental performance. So this is one of the example where we did recently some research, we published a research about overcoming strategies of ethanol production using this saccharomyces, one of the let's say industrial yeast system. So along with the factors, the chemical stresses, we have physical stresses also to be accounted when we talk about a logical LCA and also environmental assessment for assessing bioprocessing. So having said that, it says bioprocess scale up is a critical process, but loss of production performance in scaling and overcoming strategy are the keys to formalize in a modulation or competition simulation methodology. And yes, and this the last one slide is about more about Q&A to question computational scientists, how computer scientists can be helpful in bio-based refineries and tackle the question, the problems in microbial bioprocess. Here comes is one of the example where we have the biomass raw materials, and we have the product and product. But there are several technical uh, platforms, or the platform where we get the technical um, uh, products, the conversion if in processing technologies. So we get new products. We also get some hindering products. So with the help of computational framework, we expect this will be able to develop a technology or provide insight into viability of the microbial cell factory. Also, a simulation library for a range of crude enzymes. There are technologies available which say the purified enzymes or a particular specific enzyme system or bioprocess system. We're talking about development of simulation library for range of crude enzyme mixture. And um, then then also do a complete framework that can also helpful in kinetic studies, especially when talk about adding in modifying components, kinetics, and also about a perspective indicators, which is not been included in the current simulation modules, which such as chemical structure prediction, selective sampling models, and also predict biological activity in the cell in the simulation model itself. So, so with that in mind, it's a for decision-making by refund, it is necessary to develop a comparison methodology on the above point, especially the library development and this perspective indicators. So in near future, in ideal bioprocess, it will be more on the guided simulation-based method, both real-time wet lab experience, wet lab experiment with the computational-based science-based approach. Thank you very much.
All right, thank you, Vijay. And does anybody have any questions for Vijay or any ideas about how their work could help solve some of those problems that Vijay has? So just to stress that this is an interactive session and we are asking for your ideas and would really appreciate them. So anybody got a hand to show or any ideas to throw in? Okay, and let me check the involved questions. Jackie, you've got a question. Thank you. Do I need to unmute you?
Am I unmuted?
There you are, yes.
Ah, super. Thank you for that talk. I was really interested to find out what would be the key. There are quite a few different ideas that that you presented. Which was the key one that you would like the most support on? Do you think would make the biggest difference to your processes?
Well, you mean the research domain, or you mean
yes,
okay.
yeah. Which bit would be key?
At the moment, we are going to focus on two main research domains: this biopolymers and biochemicals, especially lactic acid and succinic acids from the biomass waste, and also then conversion. These roots are very well connected. When you can convert a lactic acid and succinate to the BHA in bioplastics, and then the lactic acid and succinate and other organisms has a lot more industrial value as well. So it is very much, we are going to focus on these two directions. But everything is conversion from waste material as well as the biomass, which is accessibly available in Scotland and UK. That include the marine biomass as well.
Good. Anyone else got any ideas or questions for Vijay? Let me check for hands. And let me just check our interactive questions, see if anyone's typed anything. And the chat. Okay. Neil, could you just begin to prepare for your presentation, please?
We have a question in the chat section, Anson.
Yeah, I think Benley has one question, right, Benley?
Oh, your hand thing doesn't work.
Okay, so
yeah.
Yes,
Right.
yes. For sure, the modeling is needed is more, or like, let's say, for example, we have modeling needed, especially for, as I said, existing modules available. They talk about just a purified system or the particular system. However, I'm talking about library of the crude engine when you talk about low cost system, conversion of waste materials to the biorefinery of a value added product. And also we talk about the predictive modules, you know, these, all these, let's say, how to say this, in perspective indicators that include, we can have a simulation model developed for biological activity prediction itself within the system, ongoing processing, and also the chemical structure, which will help us to see whether it is any inhibitory components being formed, or if anyway we can itself, sulfate itself is going to materialize those, and also selective model studies within the system to get to know the information, which one is the best to select, which choose with.
Thanks, Vijay. Another question has just come in to the involved chat, and that says, you mentioned a number of optimization problems to be solved. What data for those optimization problems are there?
Can you repeat the question? There's an issues. What?
You mentioned a number of optimization problems that need to be solved. What data are there for those problems?
Well, as I said, that data will be generating from our own research, you know, especially the data from the microbial processes, such as the environmental, the stresses, including this temperature stress based, and then it can have that. Let's say, for example, if you're producing ethanol system, as I explained one of the example, then we have aluminum stresses. We have let's say we have flux stresses. We also have the different enzyme mix preparation stresses. So as I said, when you talk about the process, a bioprocess, uh, from waste material to convert it to the ethanol or biobutanol or biogas, we talk about the crude system. So there are a lot more, many enzyme systems are involved. For example, in biorefinery, we can have xylenases, hemicellulases, cellulases, beta-glucosidases, xylosidases, CMC cellulose. There are many systems are involved. So systems are available to predict, but based on particular enzymes system or particular bioprocess system or particular cell factories. But here I'm talking about a library, simulation library, which is to best of my knowledge not available to that. So that is something I would like to work with the computation scientists, where we can develop a simulation library for a range of this enzyme system for biorefinery, as well as the perspective indicators, where we are able to uh, predict the chemical structures, inhibitory complex structure, and also the modules or the end product. Some cases, for example, when discover about the natural novel product, we know the process and we know the pathways, but sometimes when we add for example, a enhanced biorefinery, we add these days we use nanomaterials. So we are adding nanomaterials to the prior process system, but we don't have the knowledge what exactly changes it is doing in the system. So we know this is enhancing the end product value, means end product levels, but we don't know the process pathway, the kinetics. So that is something we can incorporate the new simulation or add up to the existing one with the help of computation scientists.
Okay, interesting. Any final further follow-up or new questions for Vijay? Let me just check. Participants and list here. All right, thank you very much, Vijay. Let's move on now to Neil.
Thank you, everyone. Thanks, Hannah.
Hello, everybody. So my name is Neil Burns. as a little bit of a departure from what you've heard about so far, we're going to go into the marine environment. So my area of research expertise and interest is looking at population health, and in a spatial context. So that usually requires measuring the numbers of species and knowing where they are, and knowing densities and numbers per unit area. So let me just get the slides to move on. So the sort of things that we tend to think about when we're thinking about sampling species or sampling habitats, exploring habitats in marine environments, is counting fish, or counting the percentage cover of corals, or even identifying individuals. So no doubt you're all familiar with counting the numbers of individual fish, and counting the numbers of different types of species. That's a sort of relatively commonly seen thing done on TV documentaries about marine science. But we might also want to know things like the size of individual fish. If we're talking like when Vijay was talking about biomass, if we want to know biomass, then we want to know the lengths of fish. We also sometimes want to identify individuals. So the picture in panel A that you can see on the screen there is a freckled hawkfish in the Red Sea, and we can identify individuals by their freckly faces, which is useful for doing behavior experiments, or even identifying trends in population expansion or contraction. So the sorts of equipment that we use to do this work, the stuff that I use is usually stereo video camera. So you can see on the left-hand side of your screen there is a stereo video camera that's built into a camera trap, much like you'll have seen again on television programs where they're taking pictures of big cats at night and these sorts of things. So this is a baited camera trap, and the two cylindrical objects you can see are housings for video cameras. You can see the same setup being used by a diver on the top right-hand side. And the useful thing about stereo video is it allows us to measure the distance away from the cameras that an object is. So the example of the fish down in the bottom right-hand side, or indeed the length of it. So getting on to some of the difficulties that we face, you can see obviously in image A, the cuckoo wrasse that you can see there. The image was taken on the south of Arran in the Firth of Clyde. It's probably not that difficult to measure the length of that fish, but obviously fish move about in the environment. And in image C, you can see that actually working out the length of that individual might be slightly harder. And in B, the fish is not kind of horizontal to the plane of the camera. So these all pose difficulties. The other difficulties that you might imagine that we come across are just counting the numbers of individual fish can be challenging. So our normal route to solve this problem is to use an honours or a master's student and sit them down in front of hours and hours of video footage and get them to count the fish. And they're quite good at it, but they take a very long time, and the fish do difficult or make your life harder because they move about. They're sometimes difficult colours to spot, and they hide quite often. So you can see as we're moving around, there's a lot of flicking of movement. The fish hide in amongst the coral. It's worth noting as well that the landscapes are not altogether easy to pick apart from the fish in some instances. So the other examples I have in amongst these videos are fish come in a variety of different shapes and sizes. So while it's easy to train a master's student in identifying fish in general, sometimes maybe training computers is not quite as easy. So other than the coral, all of the objects in this video are actually fish, but it might be harder for us to train a computer that these objects are in fact fish, let alone what species they are. The other sorts of challenges that are faced are sometimes the backgrounds are brown, and the fish that we're trying to spot in amongst that are also brown. We have objects floating in the water. So this isn't a particularly bad example, but there are fish in the background and there are moving objects in the front. And obviously these are all kind of ideal examples. They're in the Red Sea and the water quality is pretty clear. Once we start trying to do this type of work in Scotland or round about the British Isles, it becomes an awful lot harder. So what I would like to gain from this session is to get your opinions on what you think we can do and how we can use potentially AI to learn to speed up our processes. So previously what's been done is that fish have been ID'd to species level, or people have aimed to get them ID'd to species level by computer algorithms. And obviously it's an AI learning process. I'm not sure if that's the best way forward. And I wondered whether you had thoughts on maybe if we could get computers just to recognize whether the moving object that was there was actually a fish, or if the object was, and then a human participant could identify the species, and whether you think I'm aiming for too much with this. And then the other part that I would like you to consider in amongst this is it possible to have this as open source software as well? So yeah, over to the more knowledgeable people with this.
Thanks for that, Neil. Okay, we've had a couple of questions come in on the Involve system. Do you use a color palette to calibrate the colors underneath the shallow water when you are processing your video?
Yeah, so I didn't for the videos, but the still images had been through a very straightforward processing algorithm. So sometimes with the videos, we'll just color correct. We tend to set the videos up with a number of kind of starter processes. So because it's a stereo video camera, we'll flash a strobe, and the strobe allows us to sync both cameras. At the same time, we can always hold up a bit of white plastic, a dive slate that will allow us to white balance the image. So yeah, we can do.
And do you try and use ambient light source to help in brightness? So
all of the work, I'm just trying to think whether I'm correct in saying that. Yeah, so all of the work that you saw there was only using ambient light sources, other than the very nice picture of the kelp on the rock, which had lighting coming from the cameras. So I don't know whether those I don't know how much those would affect whether a computer program would be able to identify or just pick up movements better. So I don't know, maybe the whoever asked that question could elaborate on whether I should be using lighting on the camera rig rather than relying on ambient light.
Okay. Mohammed, if you've got any ideas there, drop them in the chat. A question that I had, Neil, was do you, this is the wrong word, but do you know how fish flock? Can it's not flocking, but when
so shoaling, yeah.
shoaling, thank you.
It's okay. Sorry, what was the question?
Are there like models? So do you know how different species of fish will always shoal?
So there's some incredibly interesting work on this by a chap, Ian Cousins, in Germany. So he is of the mind that it's purely speed driven. So there's lots of behavioural work done on this as well. So there's suggestions that bold individuals will go to the front, but actually there's a school of thought who think that the shoaling movement is purely a result of, so if they all move together at the same time, it's because they're all going quickly in the one direction. And you see the same thing happening in queues. So the sort of the same work that he does also informs moving people about in football venues or in say like the Olympics, the Beijing Olympics, his work was used. So yeah, they're natural forming occurrences rather than being a behavioural driven thing quite often.
Okay, thank you.
But they make our lives a lot harder. Can I flash my screen up just a second, just for the last slide? Because I would be keen, my email address is at the bottom of that slide. If anybody wants to get in touch to maybe develop these sorts of ideas further, you'd be more than welcome to email me.
Brilliant,
Thank you.
So Benji, could you get ready, please? And just to again go back to the participants and see whether there are any final questions or ideas for Neil. All right, and just going back to the involved questions. Okay, thanks very much, Neil. Benji, are you ready to start your presentation?
Yep. Let me share my screen. Yeah.
Thank you. I can see you.
Cool. Okay.
Now I can see the screen. Thank you.
Okay. Hello, everyone. Glad to have a speak here. I'm Benji Li, a quantitative geneticists and also an animal breeder at SRUC. Today, I wanted to share something with you on when genetics meet informatics in animal breeding. So firstly, what do we do? Like, what do genetics and the breeder do? And how does it overlap with data scientists or computer scientists? So animal genetics breeding is a data-driven science to understand genetic architecture of animal performance and also to select for animals with better performances in the population. So for example, we would like to select for better animals, better cows who can produce more milk in their genetic potential, or we want to choose which sheep are performing better in producing high quality wools in their genetic potential. So basically what we have is we have many animal data for their performances. And we also have their DNA sequence. All this information are stored in a big database together with their DNA sequence, performances, and their family relationship data between the animals. The data will be statistically analyzed with Bayesian or linear mixed models to get us output of animals' genetic values and also the genetic architecture, the genetic background of animal performances. So that's basically what we do. And we are interacting with data almost everywhere in our study. What is happening now at SRUC is we're doing many exciting genetic breeding research. I need to mention one thing, that's Edinburgh Genetic Evaluation Services. That's a unit within SRUC to provide UK national genomic evaluation for all dairy cows, all sheep, and the biggest beef breeds in UK. So we are working with national database, super pedigree with animals relationship, and we are doing research also to offer a hand to industry. Most recently, we are doing many genetics research related to environment. So SRUC genetics research are very interested in data-driven innovation for genetic research. We are currently getting many different formats of data. The most common one is the flat table, and we have data from animals sequencing or genotypes or epigenome data. And then we also got picture data that can be animals CT image, or for example, this one is a dairy cow's foot disease. So if there is a lesion on a cow's foot. And also we got this kind of curves that's from milk spectral data. That is from analysis of milk, helping us to know fatty acids of the milk. So basically, because we have so many different types of data, we're not only interested in quantitative genetic statistical modeling, and also we are very much involved in bioinformatics and machine learning. Machine learning is the thing in my last slide. I want to go a little bit deeper and open discussion with you guys, because machine learning is a really hot topic recently in genetics. But researchers are kind of having some difficulties right now. We need some more professionals for their help. I'll just give an example. We try to use machine learning method to predict feed intake of a cow. So that is to say, if machine learning can help us to know how much feed a cow is eating per day. This cow, not a group of cow, but a specific cow. This topic is related to feed efficiency of farms and also global warming, missing emission of livestock production. So it's a really important topic in genetics world. What people are doing now in order to collect feed intake of a specific cow is to install this kind of automatic feeder on research farms. Every time when this cow goes into the feeder, before and after the cow eats, we will know how much the feed has been consumed by this cow, and the data will be sent into a center database. But the thing is, this kind of machine is very expensive. Farmers usually cannot buy them. So what we can achieve is install them in research farm and get a few hundreds of data for a few hundreds of cows. That's not enough. So researchers are thinking about if we can use machine learning to help us. So first group of researchers are thinking, okay, can we use camera to take picture of the feed when a cow is eating? They reduce the feed every time when a cow finished eating. That picture will help us to predict how much the feed has been eaten. So this is one strategy. The other strategy is to say, okay, is that possible to use just a few hundreds of real data to predict other animals' data, even their real records are not available? So this is the idea SRUC research group is more involved in. And what we have seen now is because of the data, animals' data are quite complex. Animals can come from different environment, herds, treatment, nutrients. So we don't have very good prediction accuracy if we use a group of animals' real data to predict others. And that's mostly because of the complex data structure. Another thing is people are exploring different machine learning methods and get a headache sometimes to know which one to use. So that's the most frequent questions we have. We are very open for discussion and collaboration for more professional help. With this, I would like to thank you very much for giving me this chance. And this is my little email if people are interested in collaboration or have a talk later on. That's it.
Thanks very much, Vijay. Okay, so computer scientists in the session, please put your thinking caps on and come up with any ideas or questions for Vijay. Right, I need to just have a look at my list and see if any hands have come up, or see if anything is coming through on the involved questions. Any ideas? Just need to refresh that. Okay, so Vijay, I've got a question, and I'm just wondering, in terms of the photo taking, is any of that automated, or is there always a human required to take those photos? So, for example, that the cow's foot lesion, you know, you needed to kind of properly get it in focus and kind of get it big enough and that kind of thing. Can you imagine opportunities for, I don't know, you know, cameras sighted to be able to automatically take a series?
That's a really good question. People are doing that. So the pictures I have put down is researchers really go there and take pictures by hand. But for the feed intake, like to take pictures for how much the cow is eating. Now a group of Danish researchers, they are really set the cameras inside the farm. So as far as the cow is into the feeding area, the cow will be taking a picture. Then this is a nicer way to do this, and this is more automatic phenotyping. But some farmers, they are fond of this idea, but the others, they don't really, because they don't want to change their farms and install cameras here and there. But many farmers are like this idea because they can get much more data information from their farm. The camera, when the camera is installed, it will not only take feed intake, it can take a picture of the cow, so we know the size of the animal, because the size of the animal can be used to predict the weight of that animal. So, and it will be plenty of other information we get from installing a camera, for example. So that's an ongoing research project, at least I know in Europe. Yeah.
Right, thank you. And I know that Neil has a question as well, please, Neil.
I do, yeah. It was a really interesting talk. Thank you. I was wondering whether, I mean, this is probably something that has already been done, but given that we have a facility now to attach a number of different devices together, Bluetooth or whatever, I wonder whether you could record the number of maybe like jaw movements. So you could have a sensor attached to the cow's jaw, or even, you know, like a collar on each one, which would record movements, and then also pair that with the camera. And then so you don't need to train it to learn individual cows, because they could probably have just a little RF thing in their collar, so that the camera knows who they are.
Yeah, that's a good idea. The color thing is available on the farms, but not all the farms. So the collar was tightened in the leg, one of the leg in a cow, in some of the research farms, to count for how many activities they have, how often they drink, how often they eat, they get milk. So that's activities records have been recorded in that way. But for feed intake, maybe that's possible to have a camera together with a collar. Then the collar can not only do counting the steps, the activity, but also can take picture when the cow is eating. That's a very good idea. And then the camera need to somehow know the ear tag, so there is an ID in the cow's ear to show which animal it is. So that the camera will need to catch the information there too, in order to know which animal eating how much of feed. But that's a very good idea.
Great, and thank you, Neil Minji. A question here. So 2 of the possible factors that decides prediction accuracy are: 1, the choice of the machine learning model, and then 2, the amount of data available. So I wonder if you have any comments about those 2 things coming together.
Thank you for the question. So I would like to first bring the second factor, the amount of data. That's a really key factor we have here. So for most of the, like for milk, how much milk the cow is producing, for those kind of common animal performances, we really have millions of data. So in those parts, machine learning helps us to get predictions with very good results. But the thing is, with a linear mixed model or parameterized modeling way, not using machine learning, quantitative genetics are able to get even better results because we know the architecture, the structure of the data. So the more challenge we have now is, for the data, we don't have a lot, a big data set, maybe a few thousand of animals. That's for feed intake, we have a few thousand of animals. And when we are using our model, parameterized model, we didn't see good results. So people are wondering if machine learning is a better way to find the structure of the data as alternative. So that's for data. For feed intake, a few thousand, that's for this performance. The second is the methods. The more methods, very often genetics are trying now is deep learning in terms of a neural network, for example, and then random forest as the other machine learning method. Those are very frequently used, and what should I say? So we didn't find good accuracy there, and people are keep trying with more different methods. Usually studies from different groups have very different results in terms of methods.
Brilliant, thank you. And I've just noticed a question that's really related, but to Neil's data set actually. So this is that reflection that because you need a large amount of annotated data to train the prediction, you know, do either you or Neil have any annotated data? So that's not just the data, but the kind of metadata on the data as well, or annotated data to begin to train the machine learning systems.
Well, probably I will start for breaking the genetics, because we already have many basic data. So we have the data to train model first before getting the next of it to be predicted. Yeah.
Neil, what's your response to that?
And yeah, for in hours and hours and hours, or you can measure it in kilometers of Red Sea, in fact. So yeah, if anybody's interested in diving into that with me, that would be excellent. So yeah, there's a lot of time points recorded where fish are recorded, and also the species. But like I said, I'm not that bothered about learning getting the computer to learn the species, more just to be able to recognize the fish.
Brilliant, thank you. All right, so I think we're at the end of Binji's time, and going to come to Jackie next, please. And I will keep my eye on questions for anybody that's presented in this session, but over to Jackie next, please.
Hi there, can you hear me? Is this working?
Yes, I can see this slide.
Super. Right, well, I'm going to share this story about Selenium, the pandemic, and collective action, because I think there's an important role that computer scientists could play. Now, Selenium is quite unique. It's an essential
You just might have gone on mute by accident there, Jackie.
Hello? Is that working now? Fabulous. I'm not sure I lost you, but I'll go back to where I was. Selenium's unique. It's an essential micronutrient to humans, but selenium is also essential to RNA viruses. So our dietary selenium intake not only influences our biochemical responses to infections, but it also influences the evolution of RNA viruses. So selenium is quite important, firstly because selenium deficient hosts foster the evolution of more virulent RNA strains, but secondly there's emerging evidence that selenium status is associated with the survival during COVID infections. So I guess a good question is, where do we get our selenium from? And as a soil scientist, this is why I'm talking to you today. The answer is the soil, because selenium is not essential to plants. So if you imagine a plant and its roots is simply absorbing selenium from the soil. And low selenium soils mean little selenium is accumulated by the plants. This is why crops grown in the UK, Europe, New Zealand, Africa are insufficient in selenium for human nutrition. Now, the only behavioural model for farmers is the rational actor model. Farmers are paid for quantity, not quality. Selenium is not essential to plants, so there's no yield benefit by adding selenium fertiliser. So who cares? And that's the gist of it. Now, this creates a gap. And this gap's been known for decades. And it's behaviourally relevant now because selenium status is a risk factor impacting the outcome of COVID-19 in populations where selenium intakes are suboptimal. And as I mentioned, selenium-enriched fertilisers are actually a public health strategy in different areas of the world. It's used in Europe for over 40 years, and it's increasingly used in Africa. And 10 years ago in the UK, we developed these fertiliser guidelines for wheat. But the behavioural economics model that was applied was relying on supermarkets who made the decision. They felt because people don't really know what selenium is, there was no profit in food chain selenium. And the trickle-down effect means that there's no selenium fertiliser available for farmers to use in the UK. Now, since then, digital technologies have created opportunities for the co-evolution of ideas and people. And contrary to textbook rational actor models, people do take actions because they care. In fact, 25% of environmental management by farmers is unsubsidized. They're not paid to do it, they do it because they want to. And farmers are increasingly using Twitter to learn about sustainable soils management. As a soil scientist, I'm changing the way that I work and integrating science into these social learning networks. And today we've had a go at soil physics or soil erosion with Groundswell, and soil health focused on earthworms because farmers use earthworms as an indicator of good soil management. So I wondered, could this sort of model work for pro-social behaviours in soil chemistry? And I am going to submit a research proposal in a fortnight, just suggesting that we should just put this idea out there. And the aim is to support altruistic collective actions connecting farmers to the nutritional quality of their produce. And our aspiration is sort of developing this conversation and respecting the sovereignty of choice to participate. And this is an ideal world sort of scenario. In the real world, this open project could have all sorts of sort of effects, shall we say, and that's why I'm here reaching out for some help. Selenium biochemistry is extremely complicated and food production is politicized. You know, we've got fake news and facts which just turn off to everyone involved and kill the initiative. And this could come from narratives around the use of chemicals farmers use, which creates controversy. Then we've got celebrity opinions. For example, in America, selenium supplementation, which I've shown here, would actually be a risk to population health compared to the UK. So it just creates misunderstandings and misinformation in this connected world. So the idea that I had is, rather than just our disciplinary silo of a soil scientist working with farmers and social scientists, is there a role there for for computer scientists to help us with some dynamic network analysis, exploring how this idea that we have would evolve and develop across online communication platforms? And I've read, that there are opportunities to look at links between network characteristics and participation, how information is exchanged, and just so that we can check that Selenium's understood and that our collective actions will be safe and appropriate. It's a different sort of project. I've never suggested anything like this before, so I really welcome any suggestions. And if you'd like to be involved, then please do get in touch. And that's it.
Thanks very much, Jackie. I wonder if you might leave that last slide on screen just to help the ideation. That's great, okay. Thank you. All right, so computer scientists in the session, what questions or ideas do you have for Jackie? And please either pop your hand up so that we can unmute and hear your question, or type it into the involved technology. Right, I'm just refreshing that. One of the questions I had, Jackie, was farmers are a network anyway. Do any of the citizen observatory type of technologies or networks provide any useful methods or approaches?
I have used citizen science types approaches before. I've put out open calls and I used Farmers Weekly, print media and Twitter, and I was able to monitor what the response rate was between the two. And I found that people who were engaged on Twitter were far more likely to be involved, because it created this sort of network and we're all sharing and comparing information. So people were getting others involved and gave us much more to do and develop, which is why I use Twitter. And I've expanded that over time, and it's really quite exciting because you get to really connect with people and answer the questions that, as a scientist, you can do your field trials and then just tell people to do it, but they need to sort of integrate into that practices. So we're just making it a lot more efficient. We're just doing it at the same time together.
Yeah, and that network effect happens because you're already in a network.
Yeah, we just sort of develop it. We put out these open calls and people just pick it up and spread it across their own networks. And it is more of the innovative farmers networks, so the no-tillers and the organic farmers who tend to participate. But yeah, it's sort of a ground up kind of opportunity we thought we'd explore.
Brilliant. All right, so any questions or ideas for Jackie from anyone else? Okay. I'm just going to take a minute just to look through the questions to check that I haven't missed anything for anyone else whilst you have a think about ideas for Jackie. Yeah, there's another thought for Binji here that just came in a little bit later. Binji, there are approaches to augment data. So perhaps you just kind of do a small set and that, you know, do a small set of data augmentation on a small set and then apply that to a larger set. Might that be a possible and appropriate way to think about how you move forward?
Yes, thank you very much for the discussion here. We are trying to use a similar way like this. We are using the data we have as the kind of training annotation part, and then to get them as the training panel and to predict for other data. But then the problem is, since the current data sets is not that big, so it cannot reflect the data structure of all the other animals in a bigger population. So I think what we really need to do is to sampling more data from different environments. So our training will reflect a more wider situation of different environments. Then the training to prediction will perform better. Yeah.
Okay, thank you.
Yeah, thank you.
All right, any more ideas for Jackie or indeed for any of us before we come to the end of this session? Okay, let me just check this one. All right, well, I think we're probably at a natural end here. The questions and ideas that you have given us have been very useful. Thank you very much. And thank you for giving us the opportunity to introduce our research and our challenge centres to you, and hopefully you've picked up people's email addresses to follow up after this, but obviously the session has been recorded as well, so there'll be an opportunity to go back and pick up people's contact details. But thank you to all my colleagues for their presentations today and for their chat. And thank you to SIXA for hosting us. It's been great to be here, and hope the rest of the day is really good. So that's me bringing this session to a close. Thank you very much.
Thanks, Anna, for organizing this session.